Thanks, Art. So when Masad Dama called me on behalf of the OTS Awards Committee and informed me that Stan won the OTS Lifetime Achievement Award, I, of course, was thrilled with the news, and I immediately called Stan and informed him of this. And this was his acceptance letter, and I'll read it for you. Thank you to the OTS Board of Directors for this honor, which I am pleased to accept. To be recognized by one's peers is the highest form of recognition. So what I thought, in light of that, what would be more appropriate than for Stan to hear from some of his peers? Many of you folks in the audience who have been impacted by Stan over the years, others not here today but have been impacted for decades or in other measures by Stan, and also some pictures and voices of patients that he has impacted.
In 1989, Dr. Stanley Crooke founded Ionis Pharmaceuticals. Today, with over 3 dozen RNA-targeted drugs in development, he has built one of the largest and most advanced pipelines of drugs in the biotechnology industry.
So one of the really remarkable things about Stan is that he had a vision 30 years ago and he carried through that vision to today.
So the first time I met Stan was in 1980 when I was interviewing for a graduate student position at Baylor College of Medicine. And this gentleman walks out of the office dressed up very nicely in a, what I thought at the time was a very expensive suit. And Dr. Bush, the chairman, introduces me to Stan. And as he's walking out the door, and later on when he closed the door and I was talking to Dr. Bush, he told me to keep an eye on that gentleman. He says he's really going to be somebody that's going to be important in the pharmaceutical industry. And that person turned out to be Stan Crooke.
Another thing that he always used to say is, and probably he's saying still, is anytime you walk into the company, ask yourself, what can I do today that will help a patient, right? I think there are so many things and so many lines and so many things that he has done over the years. He has talked about things. It's just like incredible, actually. To know him is great. It's absolutely great.
Stan, congratulations on the award. I've learned a lot over the years and seen tremendous progress, and can't be more proud of what the company's accomplished and what you're getting recognized for today.
Stan, I'd like to congratulate you on the award and thank you for creating an environment at ISIS that has made so many great collaborations with my lab possible over the last 15 years.
Hi Stan, congratulations on all you have done to make Antisense technology what it is today and what it can be tomorrow. I think it is a heroic achievement to build an organization focused on the big dream of making Antisense work to help patients.
One of the memories that is most cherished in my mind were actually the down moments. And what I mean by that, Stan, is that during those times was when you really, I guess in my own view and my own understanding, you became the leader that brought everything back to focus.
Influence of Stan. He's a great mentor. He taught me the joy of, and I think a lot of us, all of us, the joy of asking and answering wonderful scientific questions. And I think most importantly, how to develop drugs. And what better purpose do we have in life than to help sick people? And it's a wonderful gift that Stan gave us for the technology to be able to do that. Congratulations, Stan. I'm so thrilled that you're being recognized for your perseverance, your brilliance, and most importantly, the dedication to the patients who benefit from the technology you pioneered.
Stan has been the absolute leader in oligonucleotide or antisense drug discovery. Absolute leader. And I certainly support him going forward, and I hope everybody else does also.
Hi, Stan. This is John Reed. Wishing you my heartfelt congratulations for the much-deserved recognition. I continue to marvel at your many, many impressive contributions to science and medicine. As a fellow fan of RNA therapeutics, Your pioneering work has laid the foundation for an entirely new platform for producing medicines for patients.
The impact that he's had on the science and medicine of oligonucleotide-based therapeutics is second to none, and it's an impact that'll be felt for years to come. Again, Stan, congratulations.
So Chief, congratulations on your Lifetime Achievement Award. Very few people in this world are privileged with the opportunity to create even a modest new drug. What you've done is created a whole sector of the pharmaceutical industry that will produce numerous drugs and help countless numbers of patients, improving their lives.
I want to congratulate Stan on this wonderful occasion of getting the Lifetime Achievement Award. I hope you are playing and winning your basketball game every day as you used to do.
Oh, hey, boss. Congratulations on your award. You know, you and I go back a really long ways, and I remember when I first met you, you were on top of the large pharma world at a very young age, very successful. And yet you chose to push that aside to pursue a big dream, a crazy idea called Antisense, because you knew if you could make it work, you'd be able to help many patients in desperate need. And you did that. And you made it fun. Also, along the way, you were able to launch a brand new scientific field called RNA therapeutics and create many important careers for young scientists, careers to be proud of. And I thank you for that. Congratulations again on your award, and as you always say now, let's get back to work. There's plenty to do.
Dear Stan, thank you for giving patients hope. We are grateful for your commitment to innovation and patience. And we are honored to know and work with you. Your dedication has given patients real hope that they can live healthier lives. And a purpose to carry on.
Your achievements will forever inspire us to continue doing great things for the sick people who depend on us.
Our technology, their hope, our purpose. Thank you, Stan. And congratulations on your remarkable achievements.
Ladies and gentlemen, please say hello to Stan Crooke.
You did me in. Give me a minute. Well, I guess we can only go downhill from here, so I better get started. Thank you very much. That was really very wonderful and kind of you. Earlier this morning, to people who didn't go to the parties last night, I spent some time talking about the history of Ionis and the history of Antisense, and that was a pleasure. A question that I was asked but never got a chance to answer is, what was the thing that scared me the most? And the answer I've given all these years always surprised people, and that was that in principle hybridization should not happen in the cell. And so for 27 years, I've been looking for the proteins that facilitate hybridization of single-strand DNA like oligos and 2' methoxyethyl's and so on, and they exist. So I wouldn't want you to think that AGO2 is the only protein that does that. There's no possibility we would be getting the effects we are getting with Antisense if there weren't some protein that were facilitating hybridization. And so it's particularly nice to follow the presentation of the AGO2 paper, because it's exactly the right question that I've been asking for many years. So in this presentation, I want to focus strictly on the present and the future. The present, because I think it's extraordinarily exciting, and the future, because the future that is taking shape and has taken shape for me over the last couple of years is very different from the future that I envisaged even 3 or 4 years ago, and has vistas, horizons, opportunities that are vastly larger than what I thought. Of course, creating Antisense technology has taken a village. And so I accept this acknowledgement on behalf of everyone at Ionis. And many people who have been at Ionis in the past and moved on and did other things with their careers, some of whom you heard from. Along the way, I've had a partnership with a wide range of senior leaders in the company who've stayed, now for many of them, their entire careers, beginning with Roseanne Crooke, Lynn Parshall, Sue Fryer, Brett Monia, Frank Bennett, Richard Geary, Scott Henry, and on and on. It's a partnership, and we've hung together and survived some very difficult times together. Of course, we've had great partners, and we have to acknowledge the patience of our investors, and most of all, we want to thank the patients that we have, the patients who participated in our trials, and the patients for whom we hope the drugs that we're developing will bring benefit. So let's focus on today. Today begins with Nusinersen. As was mentioned, 2 months after we unblinded on an interim basis the phase 3 study that was in progress, the rolling NDA was completed. On Monday, we announced that, so we're very excited about that, and we look forward to rapid approval of the drug. Nusinersen is a drug designed to treat spinal muscular atrophy, which results from homozygous loss of function of the SMN1 gene, a gene that produces a protein that's necessary to form and maintain normal mild neural junctions. And so, in effect, the muscles of the body are denervated biochemically, and as a result, the muscles atrophy very rapidly. The most severe form of the disease is also the most common in terms of incidence. It's type 1 SMA. And before today, SMA type 1 babies were born. They typically were diagnosed and are diagnosed at about 2 months of age because they don't move like a normal baby. And from that point forward, the progression of the disease is rapid, inexorable, and absolute, such that by 8 months of age, 50% of the babies are dead or require permanent ventilation. By 18 months, essentially all the babies have died. There's never been an SMA1 baby who lived beyond 2 years of age that didn't require permanent ventilation. It's a terrible disease. Type 2 and 3 are less severe, but in some ways as diabolical. Type 2 patients never walk, and many of them lose the ability to move even their fingers or arms. A number have told me that if they could just brush their own teeth, it would make an enormous difference in their lives. Type 3 patients sometimes can walk, but typically they lose the ability to walk somewhere as toddlers. So most of the patients are wheelchair fast. And Nusinersen is a remarkable drug in many ways. It takes advantage of the fact that humans have a second gene, SMN2, that would encode for the protein SMN, but for a mutation in exon 7. And so Nusinersen is designed to correct that skip splice and construct the full-length message. The SMN2 gene with the mutation will splice maybe 10% of the time correctly and give you a full-length message. And with Nusinersen, we correct that to basically the theoretical maximum. And we've demonstrated that in mice, and then in a truly beautiful experiment in the clinic in the type 1 infants, and then also in the type 2 and 3 children, looking at protein levels in CSF. So it's a remarkable drug in the sense that we see these really profound therapeutic benefits that we're seeing based on the altering of splicing of a single pre-messenger RNA. To my mind, that is the ultimate in precision medicine. It is remarkable because it appears that in a single step, we are moving from a fatal disease to long-term disease control, possibly cure, with no side effects evident to date. If you look at the history of fatal disease treatment, it's it's incremental progress over 30, 40, 50 years, and always associated with terrible toxicities. It's difficult to think of an example like Nusinersen. I go back to penicillin. And it's also remarkable because its effects are profound, and we can see that in numbers, but we can also see it tangibly in the patients that we're treating. And so I want to introduce you to a patient. His name is Cameron. He's a type 1 baby who participated in our first trial on Nusinersen. His parents took all these videos, and of course they've given me permission to show them. The first clip shows Cameron at 4 weeks of age. This is before he's been diagnosed, but you'll see he doesn't move his limbs. The next clip is 3 weeks later, just 3 weeks. In that time, he's progressed to the place where he's having very significant respiratory difficulty. You'll see that with belly breathing. From that point forward, it would be days to weeks before he would need a tracheostomy. All the clips beyond that point are Cameron being treated with Nusinersen. The third clip is Cameron after receiving his induction doses. So let me introduce you to Cameron. This is why you're in this room. Here he is. Sees bright and alert, but he doesn't move his arms or legs. 3 weeks later. This is a baby in great distress. This baby's going to die. 4.5 months on Nusinersen. Head control, which is amazing. Arms, legs. No type 1 SMA baby's ever done this. 9 months. See his feet. Great head control. Cameron's an average responder to the drug. I could show you pictures that are more remarkable. Here he is, 12 months, lifting a heavy toy. Can you imagine that? Beginning to put weight on his feet. You'll see in a minute he's really able to raise his head. It's a big deal. 20 months standing. Imagine an SMA1 baby standing. Of course, looking at himself in the mirror. 24 months, rolling repeatedly. That takes incredible strength. Several months ago, riding a horse. As I say, Cameron is about an average responder to Nusinersen. If you want to feel good, you can look up any number of patients on YouTube or whatnot, and you'll see, SM11 kids walking. So, of course, we have numbers also. This looks at a Kaplan-Meier from the initial study that we did. We're comparing it to 2 natural history studies. The most recent and the most relevant is in yellow, which is the NeuroNext study. And you can see that we're prolonging the time to either death or permanent ventilation. And we are nowhere close to reaching our median. In fact, the recent events that we've had are all accidents, just bad, we've had a couple of accidents, babies who had accidents. And these babies are living longer because they're stronger. There are many ways to measure that. You can look at motor milestones that they're achieving. SMA babies don't achieve motor milestones. You can look at it in terms of electrophysiology. There are clear electrophysiologic measurements that change. Or you can use this standardized scoring system called CHOP and 10, which is a 64-point scoring system. The typical type 1 baby comes in, as I've learned, with a CHOP and 10 score of in the 20s. And then that yellow line shows that very rapidly they progress to the place where they're totally paralyzed. The blue line is infants on Nusinersen, 12 milligrams. And you can see we're 22 points up at 26 months. That means that a number of the infants are approaching the lower limit of normal muscle performance, which is again remarkable. So right now it appears that the longer we treat, the better. We also know that the earlier we treat, the better. So we're moving to pre-symptomatic infants. We, as I said, unblinded the 110 or whatever it is baby phase 3 trial, sham controlled, and we've only reported top line, but I'll tell you that the results in the randomized trial, if anything, are more impressive than the results that we're showing you here. We will be updating data in the type 2 and type 3 patients at World Muscle, and there we will show that we're seeing basically the same phenomenon in the older children. The longer we treat, the better. We have some SMN type 2 children walking, and we have type 3 patients who lost the ability to walk, who are up and walking. I can show you those videos as well. It's, the drug works. And to date, we've identified no side effects. So I think you can understand why I'm hopeful that babies born next year with spinal muscular atrophy may never know they have the disease, other than having to come in every 4 months or 6 months and get a shot. Now, they're not going to be Olympic athletes, but I think the limits on their lives will be very different. So that's Nusinersen. Of course, we have other drugs. We have 38 drugs in development. We have 3 drugs that are finishing phase 3 almost simultaneously. Nusinersen, Volanosorsen in patients with familial chylomicronemia syndrome, and Ionis TTRx in patients with TTR amyloidosis. The pipeline that we have is comprised of 3 chemical classes. The first class is 2'-methoxyethyl. We call those 2 plus because they're significantly better performing than Canamro, and that's same chemistry, just better screening. Generation 2,5, one of the constrained sugar configurations that gives you ultra-high affinity and generates about a 10-fold increase in potency across the board for us. And then the first of the ligand-conjugated drugs, which is the series of drugs that are GalNAc. That 2'-methoxyethyl ASOs work broadly, can be administered by multiple routes of administration, work against a wide range of targets and diseases and in lots of different tissues, I think is a given today. So I really show these titles more to emphasize that I think we're trying to use the technology the way we should. That is to do important work, to tackle novel targets that have the potential to fundamentally change the course of diseases. And so we're proud of that. We also know a lot about the safety of 2'-methoxyethyls. And we're able to speak confidently about the properties of the 2'-methoxyethyls because about 10 years ago, I initiated a process that was designed to construct safety databases for each of the chemical classes that we're developing that would integrate all the safety experience from non-human primates through all of the phase 3 trials and be accessible and available to query so we could really evaluate that. And the plan was to publish. The first paper is out this year, and in that paper we looked at our non-human primate experience and our phase 1 normal volunteer experience. And I certainly encourage the other companies developing other platforms to consider constructing this and publishing their data. And so when we are speaking about the performance of the platform, I think we can speak with some confidence. And this year, that turned out to be particularly useful because we encountered, really much to our amazement, severe thrombocytopenia in the Ionis TTRx trial and with Volanosorsen almost simultaneously in the FCS trial. And so, of course, the first question that we wanted to ask, is there something that we missed? Is there something about the platform that is potentially leading to severe thrombocytopenia? And the answer is unequivocally no. This is a table from actually what is a very large report that will get trimmed down, and I will be submitting it for publication as soon as I get back to San Diego, in which we had a detailed look at the safety database with regard to platelets. Now, we have treated almost 10,000 patients with, or people with 2'-methoxyethyl's, so that's the chemical class we know best. In the database, we just principally put patients who've participated in randomized placebo-controlled trials because you need that control. So we have a little more than 3,000 patients that are available to us to ask these very specific questions. This table demonstrates that none of the 16 ASOs that contribute to this, to 15 different targets, were we able to see at any dose at any time severe thrombocytopenia with the 2'-methoxyethyls, and obviously different chemistries behave differently. So we are confident that it's not a platform effect, and in fact I think now understand pretty much what happened in the FCS trial and what happened in the TTR trial. Very different things and very unique to those two patient populations, and we'll be publishing those data after we've got a bit more work to do. But we feel very confident that we're in control of the platelet issue that we encountered earlier this year, and we would not be here were it not for the ability to analyze the data and be confident of our conclusions. As I said, Generation 2.5 is a better group of drugs than the best that we had before, which was Canamro. This is an apples-to-apples comparison. These are 4 drugs to 4 different liver targets that are, the protein is secreted into blood. These are all 4-week normal volunteer studies with the same 4 dose levels. And so we see anywhere from 2 to 4-fold increase in potency. And importantly, as we've understood more about the mechanisms of the pro-inflammatory effects, we've imposed some counter screens that allow us to screen out the more pro-inflammatory ASOs. And so we've basically taken flu-like syndromes to no longer an issue at all and moved injection site reactions to essentially de minimis levels, which are important steps in terms of tolerability. Probably the most potent 2'-methoxyethyl ASO that we've seen is the glucagon receptor drug, which is in phase 2 trials right now. This is a drug that we target to end-stage diabetes, where there's still a tremendous need for better treatment. We did an interim analysis early in the year of this 6-month study, and remarkably, 50 milligrams a week gave us more than 1% reduction in hemoglobin A1C. Looking at the blinded data in this study, my guess is when we unblind the whole study late this year, we'll see probably a little better than what you're seeing here with regard to the potency. So screening and good screening matters and can make a big difference in the performance of your drugs. With Antisense drugs, this is with small molecules. The Generation 2.5 chemistry, as we call it, that we use is CET. It increases affinity per nucleotide unit substantially, and so we see a very substantial increase in affinity and about a 10-fold increase in potency. And so the place that we're using the CET chemistry is principally to get better pharmacology in some of the tissues that accumulate less of these ASOs, like skeletal muscle and cancer. We have now experience with 4 Generation 2.5 drugs, I guess clinical experience with 3. The first 2 are cancer drugs, STAT3 and androgen receptor. And with STAT3, we saw some interesting activity, and with the androgen receptor, we're seeing interesting activity as well. These are 2 patients in the initial trial who had very advanced prostate cancer that flunked 7 or 8 regimens. So any kind of response of any sort in a patient like this is important. And we saw substantial reductions in PSAs and comparable drops in CTCs. So our sense is that we are probably seeing significant improvements in pharmacology in some tumor cells, and we're also seeing, I think, improvements in pharmacology in skeletal muscle. We have 4 of those drugs in development today, and we'll be converting the pipeline to more 2,5s over the years. Brett will be talking about our work in looking at ligand targeting of various tissues. And of course, what we're all focused on is GalNAc, which has made a tremendous difference. It has given us across the board, as Brett will show you, now we have, I think, 4 examples, about a 30-fold increase in potency with an ID50 dose in man of around 5 milligrams a week, which is a remarkable number, which of course then means the injections are really not noticeable. ISRs, tolerability goes way up. And we have a lot more flexibility in terms of dose schedule and so on as well. So this is a very fundamental advance for the field. And it was made initially at ISIS many years ago by Mano, whereupon I killed it. And so Mano, thanks for correcting my mistake for me. We have 8 of those drugs in development today. With our drugs, in contrast to siRNA, we don't see an increase in concentration in the liver. We see a change in the productive distribution to hepatocytes. And then, of course, we can combine the GalNAc or whatever ligand exchange ASO with a generation 2.5. And in principle, that ought to give you a 10-fold increase in potency. We have our first generation 2.5 GalNAc that we hope to have ready for development this year. And again, in animals, we see about a 10-fold increase in potency. So the animals then predict that we ought to be thinking about ID50 doses with this molecule in the range of about 1 milligram a week. If you can imagine treating a patient with 50 milligrams or 75 milligrams a year, kind of neat. GalNAc is not the only ligand that has interesting properties in terms of improving delivery to specific tissues. Palmitate is another molecule of interest to us. It appears to increase distribution and potency in skeletal muscle and cardiac muscle. These are tissues that we'd like to work in, particularly as antiarrhythmics and for congestive failure and so on. We'd love to do a little bit more better pharmacology in the heart. And then finally, I've been interested for some time in resolving what I consider the final barrier to Antisense, which is that it's principally a tool to reduce target RNAs and target proteins. And that means basically we can only really generate most of the time antagonist type drugs. And so our thought was, can we begin to think of ways to make agonist type drugs, actually increase the translation of specific proteins. And here we benefit from advances in RNA biology that over the last few years have shown that about 90% of the messages in mammalian cells downregulate their own translation, and they do that either with sequence elements or structural elements in the 5' UTR. And so this is work from Liang in my little group, was published earlier this year. The first solution that evolution hit upon was pretty simple. Insert a fake translation initiation site in the 5' UTR, and that reduces the efficiency of the ribosome initiating at the right ORF. And so our idea was, we'll just take a fully modified ASO, bind to this upstream ORF initiation site, and result then in the ribosome being more efficient and sitting down on the right ORF. Sounds simple, turned out to not be, but it does work. These are two figures from the paper. On the left, you're looking at human cells and one of our favorite proteins, RNase H1. You can see we get about a twofold increase in the production of RNase H1, and that is sufficient to give you a phenotype. A 50% increase, as someone else suggested yesterday, is probably sufficient for most of these downregulated messages to to give you a phenotype. Then we can also do it in the mouse cell, and on the right there, you're looking at a liver, a target expressed in the liver of the mouse after systemic administration. And so we can take upstream orphs, and they're upstream orphs in about 50% of the messages, and most of them are functional, and use them to selectively increase the translation of specific proteins. The second mechanism gives the same outcome, but is an intrinsically more interesting mechanism, and that's the insertion of structure near the 5' cap, and these are called TSEs or TIEs. And it's thought that they work because they disrupt the formation of the initiation complex, principally the single-strand RNA binding proteins. And so we designed a bunch of oligos, and it turns out the 3' side of the stem, as you would expect, is the place where we get good pharmacology. And we've now shown that the mechanism is probably right. That is, we see greater binding of initiation factors once we disrupt that structure, move it out. And this works too. On the left, again, it's human cells. And this is from the paper I think we just submitted yesterday. What you're looking at here is LDL receptors. To put that in context for you, that's about the maximum increase in these cells that we can see with our best PCSK9 inhibitor. And then we can also do it in the mouse with a liver express target in the mouse. And so we have these two mechanisms that we think are very robust that will allow us to upregulate the translation of somewhere between 80 and 90% of the messengers in the cell. So we're very excited about that and incorporating this into many of our therapeutic programs now. So the present, to my mind, is certainly very exciting. We have drugs that are working and making fundamental changes in diseases, not just Nusinersen, Volanesorsen, factor 11Rx, and many others. We know that the potency, even for the same chemistry, is getting better. We are confident that we have an acceptable safety profile, and we're confident that we are resolving the platelet issue that bothered us with Volanesorsen and Ionis TTRX. And we'll get the data published so you can judge for yourself. We know that we can use multiple routes of administration. Generation 2.5s appear to be working in man. Safety profile so far looks good. So that broadens the horizons for us. GalNAc is a game changer, and combining GalNAc-2 with generation 2.5 is an opportunity to get to places in terms of potency where I don't think drugs have been. And so we're very excited about the present, and we agree that this is a moment of inflection for the technology, and we're delighted to be a part of it. Now I'd like to focus on tomorrow, and I hope to share with you some of the tremendous excitement that I feel about where the field is moving and the opportunities to tailor-make drugs that are vastly better than what we're doing today. I think many of you know that for 27 years now, I've directly led a lot of the science that we call core antisense research, that is designed to understand the molecular mechanisms by which antisense drugs work, and then use that to design better drugs. This is the group that works with me. It's an incredible group of people. And of course, we benefit from being in Ionis, and so we get contributions from medicinal chemistry, screening, pharmacokinetics, all kinds of places. So we're certainly not alone. But I'm going to just show you work that these people have done primarily. And what I want to do is introduce this notion to you. And that is that to understand the molecular mechanisms of phosphorylthioate containing ASOs, it's now essential that we understand two codes. Not only is it essential, I believe that the progress we've made opens up opportunities for medicinal chemistry that broaden the scope for advances in the technology by orders of magnitude. And I'd like to try to convince you of that. So the codes, well, of course, nucleic acid code. I think we've done a good job of understanding the nucleic acid code. I think the SAR from the oligo side is very well understood. And we have a pretty good understanding of the SAR from the RNA side. We still need to do better at structural characterization in cells, and we are making great progress at being able to structure map all RNA in the cell. And there are some questions about some of the more typical post-transcriptional modifications of RNA and how do they affect ASO activity. But by and large, I think we're in a very good place in understanding this language. The second code is the amino acid code. And we've been thinking about this code for only 2 or 3 years, and I'm tremendously excited about the progress that we've made. I think we've made a substantial amount of progress in understanding the SAR of protein interactions from the oligo side, and we've got a lot more to do, but we're beginning to understand the SAR from the protein side as well. And there again, we benefit from tremendous advances in understanding the structure of RNA binding proteins, for example. So why do I say that? Because proteins determine the fate of phosphoryl containing ASOs everywhere. Inject it in the subcutaneous space, what goes on in blood, what happens at the endothelium, what happens in the interstitium, what happens at the cell surface, and what happens in the cell is defined by proteins and protein interactions. And this work that we've begun, of course, is built on a platform of knowledge that we've worked on for 27 years. And so we begin with a lot of information. And we know, if we think about an RNase H1 ASO, that H1 is limiting, both in rate and in extent. And we know that matters. We've sorted out a whole bunch of issues that we thought might matter at the start of the endeavor, and identified just the few that really do matter, like RNA structure. We even have worked out, and this is a paper Tim Vickers and I published a 3 years ago, the rate constants for each of the steps in each of the important molecular steps in an ASO activity having to do with activating RNase H1. And we know that all we do with a really good RNase H1 ASO is double the intrinsic degradation rate of that RNA in the cell. And if we find one of these multiple repeats, we can take that up to 3-fold. So that's the limit. That is the playing field that we're on. And so we take advantage of all this work as we think about how to understand these protein interactions. So we began asking questions about intracellular distribution. What are the proteins inside the cell that bind to phosphorylthioate-containing ASOs, and what do they do? We now have moved to the cell surface, and I won't have time to talk about that, but I think we have a pretty full catalog of the proteins that bind to the cell surface as well. And we've identified a number of proteins that bind. We've identified proteins that appear to be responsible for migration of ASOs in various places. If we move those proteins, the ASOs move with them. We've identified a number of proteins that appear to be inhibitory. Several of them appear to compete with RNase H1 as a mechanism, and quite a number of proteins that appear to enhance ASO activity. And we're answering questions about how. Moreover, for transfection, free uptake, and for electroporation, we now have a full catalog with kinetic descriptions of where ASO is in the cell at any given time. And this cartoon will be in a paper that's in press in Nature Biotech. But what it does is it says some important things. It says we know the places where ASOs go, both in punctate sites as well as diffused ASOs, both in the cytoplasmic nucleus. We know the time they get there. We know how long they stay there. And we have a very good sense now of where we want our ASOs in the cell and where we don't. We have identified a number of sites where they're clearly inactive, and a whole, and much more importantly, a set of sites where we seem to have enhanced activity. And we have this worked out for transfection, free uptake, and for electroporation. And then we can take that information and begin to put that in pharmacological context. And so here we're looking at onset of action, duration of action, and a maximum effect of two ASOs, one to a mature message and the second to a nuclear retained RNA, and figure out where the ASO is when these things start happening. And so that begins to tell us again where we want our ASOs, and then if we can manipulate that, we can make better drugs. And the breakthrough in this endeavor was made by Liang when he developed a biotin capture system to collect the intracellular proteins that bind to phosphorothioate-containing ASOs. And when I began this work, my worry was that we'd end up with 1,000 or 500 or some ungodly number of proteins. It turns out it's 52. Now, the assay would miss proteins that have low affinity, and it would miss proteins that are very low abundant. But since we're interested primarily in bulk changes in ASO location and activity, I suspect that this is the cast of characters that we need to be focused on primarily. We'd also miss indirect interactions, but this at least gives us a starting point to identify the key proteins. It begins to give us preliminary structure-activity relationships right away. For example, you know immediately from this assay that polarity of the 2' modification matters to proteins, which is remarkably interesting. The proteins we identified, many of them are not surprising. A lot of nucleic acid binding domains there, chaperone proteins, and then a few surprises along the way. We've identified proteins that seem to be responsible for moving ASOs in and out of one organelle or one spot or another. We've identified a set of proteins that seem to reduce ASO activity, and we've identified a set of proteins that increase ASO activity, and a larger set of proteins that bind that seem to have no particular influence on ASO behavior. And so our job, once this was done, was to begin to sort out these interactions in real biochemical terms and to understand the meaning of these interactions. The good news is that we only have 52 proteins, but the bad news when you start thinking about converting that to numbers is you have 52 proteins. And it gets pretty daunting to think about 52 proteins that you have to purify, renature, generate antibodies, 4 or 5 deletion mutants, and pretty soon you know that you're going to spend a lifetime. And so I was a little despondent at that point about whether we were going to be able to convert this to numbers. And Tim Vickers then took that challenge on, and this is a paper that just came out. And what he did was modify the nanoluke assay to look at oligo protein interactions. And it's an obvious sort of thing to do, but just to put an alexidai either on the 3' or 5' side of an oligo, and then you can put your luciferase in the amino terminus or the carboxy terminus of the protein. And this assay has incredible advantages, and you'll see, because I'm going to show you data that Tim has generated in the last 9 months or a year, single guy, you'll get the idea that this assay has real benefit. What's neat about this assay is it's extremely rapid. It gives you very good binding isotherms and high quality data, and we've confirmed the validity of these numbers using other binding assays. It doesn't, of course it will work with a purified protein, but you don't have to. It works in cell homogenates. You can select your proteins without purifying. You don't have to renature or denature, renature. You don't even need an antibody. If you don't have an antibody, you can use the antibody to luciferase to pull your protein down. The other interesting feature is that the intensity, that is the amplitude of the signal you get, is proportional to 10 to the 6th distance from the alexidai to the luciferase. So if you think about an oligo being, if it's linear, about 100 angstroms long, you can even see, you can begin to get a sense of whether the oligo is binding the 3' or 5' end closer to the luciferase or not. and you can get a sense of the distance from the luciferase. It does have some limits. You can't do this with very large proteins. 70 kilodalton is about the largest we can work with. And the slide says it doesn't work in live cells. We did get it to work just last week a little bit. But Tim has a new assay called the nanobit assay, which it lets us work on big proteins and in cells. So we're now getting real-time images of our oligos in cells. And that paper will get submitted here before long. Let me show you some data. This is the way it looks. This is a protein that we've become extremely interested in. This is a perispeckle protein, and we think it's involved in lots of things ASOs do. P54. And so in this case, Tim put the luciferase on the amino terminus of the protein. I think this is a 3' Alexa. But we have it for the carboxy terminus and the 5' Alexa as well. You can do direct binding or you can do competition. So that means you don't have to put an Alexa dye on all your oligos. You can have an Alexa dye on your primary oligo and then compete. This is a direct binding assay. You see you get very nice isotherms both ways. And there's a message here that I'm going to walk you through. There are quite a number of proteins, not all, but a good number, that are able to discriminate between relatively more hydrophobic 2' modified ASOs and more hydrophilic. That is, many proteins prefer more hydrophobic 2' modifications. So the more hydrophobic modifications, which are things like c-et and fluoro and LNA and so on, bind to more proteins, and the proteins they bind to, they bind more with greater affinity. So the next question was, do proteins differ in their appetite to bind an ASO? And so this is another figure from that paper. What Tim did was look at 8 different proteins. So these are all oligo-binding proteins. They're all in that 52 proteins that we found. And then we asked, how different is their appetite to bind to this particular oligo, just depending on the protein? And I think this is a 2'-methoxyethyl, but I can't remember. But I mean, the main point is there's about a 4 order of magnitude difference for this particular oligonucleotide just in the 8 proteins that we looked at. So proteins are discriminating between the kinds of oligos they interact with and the chemistries. This is a table that just makes that point with numbers. Again, this is a table that expands pretty much every day. But this is the list of 2 or 3 months ago of all the proteins that Tim had looked at. And he's also looked at a lot of deletion mutations. And at the top there, you have affinity constants or dissociation constants for fluoral, c-et, and Mo, all the same length, all the same sequence, and characterization of the proteins in terms of domains and so on. And in the red there, you can just follow the KDs, and you can see that these proteins have a preference for the more hydrophobic 2' modified ASOs, and it's about a 2 order of magnitude. Now, the next question we asked was, what about charge? And here's where I think it becomes very interesting. What we did, and our neutral backbone that we're using is what we call MOP. And so the chemists put either a single MOP in those positions marked with X's in this sequence, or they put 2 MOPs in that sequence. And I think the easier one to see is the one where we put 2. And you can see there's a really marked difference in affinity just by inserting 2 neutral charges. And the effect on this particular protein, which is nucleolin, one of the most important proteins. Remember that protein. P54 is a good one. Nucleolin's another one you want to remember. If you just look at nucleolin, the insertion of 2 neutral backbones produces a very substantial change in affinity that is influenced by position within the oligo. We can also look at it from the protein side, and Tim has looked at a whole bunch. And what you're looking at here is 2 different proteins, LA, which is an interesting protein, P54, which is a very important protein. Both of them have RNA binding domains. And so we're asking, if you get rid of one or two of these RNA binding domains, what happens to binding, and does the chemistry matter? And as you might expect, different chemistries behave differently, and so we have our first evidence that even within the same protein, not only is affinity higher, but the site of binding, depending on the 2' modifications, may vary. Finally, sequence, and this is, again, probably I shouldn't have been surprised, but I was. Sequence matters tremendously. We're looking at just 3 proteins, La, P54, and nucleolin. And different proteins discriminate sequences differently. Some are very promiscuous and others are very specific, or relatively specific. This is all the same oligo design, just different sequences. And what you can see is remarkably there's a significant variation within each protein in terms of the affinity as a function of sequence. We haven't really identified any motifs. It's not surprising that 3 or 4 Gs in a row will give you some strange behavior. So what I tried to show you in a very brief way is that proteins matter. These interactions play a seminal role in the performance of ASOs, and the opportunities for medicinal chemistry are enormous because very minor modifications give you substantial changes in behavior. We now, as I said, have a catalog of, I think, the main proteins at the cell surface. We think the decision to enter productive pathway or not is made at the cell surface. We think the main interactions are with proteins at the cell surface, though the lipid milieu makes a difference, and we'll have a couple of papers coming out about that. We've identified the intracellular proteins. It's a manageable number. We have an assay that allows us to do it. And we've begun to identify the really important proteins and the sites in the cell where we want our oligos. So we have a great medicinal chemistry opportunity. We have a fabulous set of targets, and we have the assays and the abilities to address the questions that we want to ask with them. That's called a great opportunity, and that's the future that I'm entertained by. Again, nucleolin turns out to be a really important protein. So structure-activity relationships, number of phosphorothioates, hydrophobicity of the 2' group, polarity of the 2' group, sequence, charge. There are some proteins that are very promiscuous. They'll bind to almost any phosphothioate-containing oligo, but most are pretty selective. We're beginning to understand the structure-activity relationships from the protein side. RNA binding domains and nucleic acid binding domains, not surprisingly, play a big role. The 2 highest affinity proteins are nucleolin and the single-strand binding protein. Nucleolin probably is the important one. SSB1 probably doesn't play too big a role. Nucleolin binding correlates with potency. It also correlates with some adverse events, and so we'll be telling you about that in due course. And now we're moving on to really understanding the binding site, stoichiometry, and so on. It's a very exciting time for us. And our medicinal chemistry program is now focused to a significant degree, no longer on nucleic acid interactions, but on our interactions with key proteins. And that's where we think the real advances in the coming years are going to come from. So we have the key proteins, we have the pathways, we understand what seems to be happening with regard to subcellular distribution, and we have a manageable, achievable medicinal chemistry challenge in front of us. And so we think that makes for a future that looks quite different from what it did to me 2 years ago, and opens a set of horizons that I really couldn't have imagined 4 or 5 years ago. So we're really excited about it. So in conclusion, we think Antisense is at an inflection point. We think Nusinersen will be rapidly approved, and we are confident that we're going to fundamentally change the course of that disease. That is worth the price of admission. And we've got lots of good drugs. And we think that tomorrow will be driven, as it should be, by what's going on in the lab, and what's going on in the lab is really exciting. Thank you very much.
we have time for a few quick questions. I'll ask one. Given the great safety of Nusinersen and the very encouraging early results, are you going to continue dose escalating, or do you think the current dose is really the maximal therapeutic effect?
Well, we'll see with time. There certainly isn't a dose limit with regard to toxicity or ability to deliver. If you look at, I think we published this now, or if not, we've presented it. If you look at the splicing that we got in the autopsy study in the infants, it's at the theoretical limit. We got just about what you'd expect if you were including exon 7 100% of the time. So in principle, as long as we continue to see that, there's no reason for a higher dose. We thought about it at the time and decided there's no justification for it. Who knows what may happen with various tolerance mechanisms that may be protein related or so on. If we start seeing some of that in some of these longer term, we've got now some infants that have been treated for 3 years, and the effect is not waning, it's becoming more pronounced. But if we start seeing that, then I think first step will be to go back and use a higher dose. We're as much interested in making sure that the drug gets used pre-symptomatically as anything. We think the key here is to treat early, and there will be data about that, and that will be reported one of these days. But so we're focused on earlier treatment. We're focused on, and we think if we can get them earlier, there's no reason for them to know they got a disease. And then we'll see how it goes over the next few years. This is a drug, so tolerance should develop in some way, and we should be ready to go forward with higher doses if we need to.
Stereos, and then in the back.
Congratulations on all your career, especially on this drug for the newborns. And I think it's a great case of how data should drive successful drugs. The question I want to ask you is about the RNA binding proteins you've just identified. Given they are there, and we've heard a lot about long non-coding RNAs and so on and so forth, have you tried to see what natural transcripts might be implicated in their functions and how these might be influenced by your drugs?
Well, if you get rid of all your phosphorothioate, you need 6 or 7 phosphorothioate. So this information is going to be relevant at some level to double-strand RNA as well. You really lose meaningful binding. So I think the binding to natural substrates is driven by many properties that the ASOs don't have. I think what the phosphorothioates do is they give you, a slightly more hydrophobic character of the phosphate, and that encourages then an interaction with the protein. If you think about it, based on what we've seen, in contrast to what I would have expected, clearly the long-range interaction is charge-charge. But all the real intimate interactions in the protein is got to be base stacking on amino acids. It's got to be interacting with metal pi interactions and that sort of thing. And so I think all the phosphorothioate does is reduce that charge repulsion, and then you take advantage of other properties. And that's what we're working on now, is to see if we can really understand the character of those binding interactions at a fine level of detail.
Thank you very much.
Hello, I'm Hai Fangyin from Tianjin Medical University, China. Congratulations for the award. And I'm really interested in your fascinating study published early this year in Nature Biotech for using antisense oligonucleotide targeting the arm-stream open reading frame. I think it's great proof of concept demonstration for using antisense. But my question is, how do you generalize this approach? As I know, probably there's not many genes having an upstream open reading frame. Do you get my question?
I didn't, I'm sorry.
Sorry, my question
I understood everything till the question.
my question is, you're using the Antisense oligo to targeting the upstream open radioframe for modulating the translation. By which way, this way you can up-regulating the translation. But as far as I know, there are not many genes having such feature. So how do you generalize this approach?
No, that's actually not my understanding. If you look, about 50% of the messages that we've looked at have upstream ORFs, and most of them are functional. And about 30 to 40% of messages, then we'll have TSEs. And we developed an algorithm that we can use to help us identify the functional TSEs. So in fact, and we've now looked at, I would guess 20 messages in some depth. And all but one or two had functional 5' UTR translation suppressors, either TSEs or ORFs. So I think it's actually fairly generally applicable. And the real challenge is optimizing hybridization, because you need enough to bind, but you also have the ribosome has to peel that off to read through. So I mean, that's what has taken the time. I think it's fairly broad. It's not going to be every messenger, but it's fairly broad.
Is there any database for showing such genes? I mean, is there any database for showing the messenger RNA having such 5' UTR, they have this feature?
None. I mean, there's no teleologic reasoning why most messages have a repression of their own translation. I can make up an argument that might be necessary for many proteins so that you could quickly upregulate their amount in times of stress. But it's fairly straightforward to identify the functional ones, both TSEs and the ORFs. It's not trivial, but it's fairly straightforward, and it's fairly broad. So we'll see over time how much therapeutic gain we get. I think what we have to do now is get these things in therapeutic programs and really ask, are we going to see a meaningful benefit in these models? But we think we've got a broad tool.
Okay. Last question.
Congratulations, Stan, and I must say that I've used the papers of your company, and all the work I've done has been based in part by work published by ISIS. So my question, though, is about RNase-H versus blockers, and it's kind of surprising after all these years that two drugs that are being approved are blocking drugs, this Sarepta drug and your drug, and it's sort of been assumed that when you want to turn off a gene, you want to cut with RNase-H or RNAi, but when you want to modulate it some other way, like splice blocking, you use a blocker. Do you see a role for blocking in inhibiting gene expression?
Well, I think what you've identified is that there are multiple mechanisms we can pick from, and if you're thinking pharmacologically, you'll pick the best mechanism. I still think that if you want to reduce a target, the best way is RNase-H1 or AGO2. I think the catalysis matters. It is possible to inhibit translation. It's been done. It was done 27 years ago. But in our hands, the potency and efficacy are substantially lower than what we see with an RNase-H1 ASO. I think for other purposes, whether it's increased translation, as we're doing with the ORFs and the TSEs, or it's driving splicing, then you're not attempting to reduce, you're attempting to do something different, and there I think the different chemistries and different mechanisms are available. One thing I hope organizers that you think about possibly Cameron being the icon for my acceptance of this award. It really matters to me to be able to share Cameron with you today. It's very special. But anyway, okay, did I answer your question? Yeah, I think you've got choices. And you make rational choices in pharmacological context, and you'll get good answers.
And looking at the time, Art, I'm going to ask if you can ask your question of Stan later, and we'll move to the award presentation, and then we have a break.
Thank you very much, everyone. Very nice of you, Bill. Very touched.